We all learned this about in biology class, right? DNA is the blueprint of life. It's this master plan, the architect holding all the instructions for building, well, for building us. It's a really powerful and simple idea. But what if it's not actually the whole story?
So this elegant little diagram here is called the central dogma of molecular biology, and it tells this really clean, linear story. Information flows from DNA, it gets copied to RNA, and then that RNA is used to build proteins. For decades, this was it. This was the model for how life works. DNA is the boss, RNA is just the messenger, and proteins do all the work. Simple.
But what if that super clean one-way street is actually more like a chaotic city intersection with shortcuts and U-turns and all sorts of complex traffic patterns? What if RNA, that humble messenger, is actually so much more?
Okay, so to really get the revolution that's happened, we first have to understand the original rules of the road. So let's do a quick breakdown of this classic model, the central dogma, because it is our essential starting point.
So the core idea, which was first put out there by Francis Crick, is that all this genetic info flows in just one direction. It goes from the master blueprint, the DNA, to a working copy, that's the RNA, which is then used to assemble the final product, a protein. It's a one-way information highway. No exits, no turnarounds. Now, this information highway has two main processes that make the whole thing go. One is all about copying the plans, and the other is about turning those plans into an actual physical thing. All right, let's look at these two steps. Think of transcription as just copying notes in the same language. You're going from the genetic language of DNA to the nearly identical language of RNA. But translation, now that's a whole different ballgame. You're actually converting or translating that information into a completely new language, the language of proteins, to build the actual working parts of the cell. And even that copying process isn't super clean at first. The initial draft of RNA, called pre-mRNA, is kind of a mess. It has the useful instructions called exons, but it's also got a lot of what was thought to be junk, these things called introns. So the cell acts like this master editor, snipping out all those introns and stitching the useful exons together to create a perfect final draft of instructions. So for a long, long time, that was our understanding, a nice, neat process. But science just loves finding those little exceptions that break all the rules. And in biology, that exception turned out to be a total game changer. I mean, the word dogma itself implies something that's true without question. And if there's one thing science is really, really good at, it's questioning the unquestionable. And this is where the retrovirus comes in. Probably the most famous example is HIV. Now, unlike us, its genetic material is RNA, not DNA. And to take over our cells, it has to pull off a trick that the central dogma said was absolutely impossible. It has to write its RNA code backwards into our DNA. And there it is. That's the crack in the dogma. The process is called reverse transcription, and it's literally the information highway flowing in reverse. The virus uses this special enzyme to turn its RNA back into DNA, and then it hijacks our cells. Suddenly, that one-way street became a two-way road. But this one exception, it was just the tip of the iceberg. The real revolution wasn't about information flowing backward. It was the discovery that RNA isn't just a messenger at all. Nope, it's a powerful manager, a regulator, a crucial player in pretty much every single thing a cell does. Okay, get ready for this number, 40%. That's roughly the percentage of long, non-protein coding RNAs that are found specifically in the brain. Now, if RNA was just a simple messenger, why on earth would our most complex organ be absolutely overflowing with these weird molecules? And that brings us to the real star of the show, the discovery that flipped genetics on its head, non-coding RNA. These are RNA molecules that never, ever get turned into proteins. For years, scientists dismissed them as useless byproducts of junk DNA. But now we know they're actually critical regulators. They are the true conductors of the entire cellular orchestra. And man, what a list of jobs they have. Non-coding RNAs act as scaffolds to build complex machines. They help form little compartments inside the cell. They fine-tune which genes get turned on or off. And they even control the structure of our DNA itself. The simple messenger is actually a master multitasker.
So why does all this matter? Well, nowhere is the power of RNA more obvious or more critical than in the human brain.
Just think about it for a second. Your brain has to react and adapt at lightning speed. Your neurons can't just wait around for a signal to go all the way back to the DNA in the nucleus to make a new protein. They need fast, local control right there on the spot. And RNA, with its flexibility and speed, is the perfect molecule for that job, allowing for the kind of on-the-fly regulation that's absolutely essential for learning and forming memory. And when this incredibly complex RNA network goes wrong, the consequences can be just devastating. This table here just shows a few examples of how specific long non-coding RNAs are directly tied to major neuropsychiatric and neurodegenerative disorders. The connection is becoming clearer every day.
And the scale here is just mind-boggling. A tiny change. We're talking a single letter typo in one of these non-coding RNAs can mess up its function enough to be linked with really complex conditions like autism and schizophrenia. It just goes to show you how incredibly essential and powerful these molecules really are. So where does that leave us? Well, we've gone from a simple, straight-line model to this complex, dynamic network where RNA is a central player. This isn't the end of the story. Really, it's the beginning of an incredibly exciting new chapter in biology. Right now, researchers are tackling some massive challenges, which are really the next big frontiers. They're trying to catalog the mind-boggling diversity of all these RNAs, predict how their 3D shape decides what job they do, and map out the vast network of molecules they team up with. Each one of these is like a whole new universe of discovery, just waiting to be explored. This quote really just sums it up perfectly. We've gone from seeing RNA as just a passive go-between to understanding it as a central, active force in the cell, building, catalyzing reactions, and regulating life itself. In the end, the story of RNA really shows us that even the most fundamental rules in science are just our best guess at the moment. They're just waiting for new discoveries to show us a deeper, more complex, and honestly, a way more fascinating reality. It really makes you wonder, what other biological rules are just waiting to be broken?